Hello, I'm Dr. Trevor Stammers, and I'm Director of the Centre for Bioethics and Emerging Technologies at St Mary's University. And this is the first of a series of A-level talks for non-scientists looking at the ethics of genomic editing. And this first one is on genetic determinism. Well, there can be no doubt at all that genomic editing is here to stay. And the recent advances that have hit the headlines are just the base camp of the Himalayas that we're likely to scale in the coming years. Already there have been some considerable advances in terms of cures, and this is Layla Richards, one of the first in Britain, in 2017, who was cured of her leukemia by her bone marrow cells being genetically edited. And as far as I know, Layla is still alive and well. But of course, wherever there are great advances like this, there are always great perils to be avoided as well. And this is illustrated by this slide showing Professor He Zhuanzhi announcing the birth in 2018 of twin girls, which he had genetically edited to be resistant to HIV infection. And contrary to what you might expect, this announcement was greeted by virtual universal condemnation by the global scientific community. And this was for several reasons. First of all, Professor Hay had edited perfectly healthy children. They didn't have HIV and they weren't at particularly excess risk of getting it. However, the editing has left them more susceptible to getting influenza, and that is much more common than HIV and can, in some circumstances, of course, prove fatal. And it's very common in China. And furthermore, the editing process that he used was incomplete. So only some of the girls' cells were edited, but some were not. And he was subsequently denounced by his own government, and I think is currently in prison for what he did, although it is highly likely that the Chinese government were fully aware all the time of what he was doing. But another key difference between what Professor Hay did and the cure of Layla Richards relates to the difference between somatic and germline editing. Now, somatic editing, as this slide shows, targets genes in specific types of cells in the body, such as the bone marrow, and these changes are not inherited by future generations. Whereas what Professor Hay did with the twins was germline editing. And germline editing in general targets genes in gametes, in sperm or egg cells, or in very early embryos. And this alters, therefore, the genes in all of the cells of the body, or at least it attempts to. Obviously, Professor Hay only managed some cells in the twins, but the aim is for all to be edited, as well as the germline cells. So these changes are inherited by future generations. So if you make a mistake, it's not only the persons like these twins who were edited who may suffer the consequences. Now, another important distinction, just while we have a summary of the science, is between a gene and the genome. And a gene is a sequence of nucleotides or particular chemical molecules which code for a protein within a chromosome in an animal cell. A genome includes both the genes, the coding regions, and for non-coding DNA, the double helix of DNA, and the genetic material that's found in other parts of the cell, such as the mitochondria and the power stations of the cell, and chloroplasts in plants. Now, this is just a sort of quick summary of the science. The structure of DNA is like a spiral staircase with a couple of handrails, one on either side of the treads, which are formed of these chemicals of the code, in which T always pairs with A and C with G, as in the middle illustration here. And proteins are made when the two strands of the DNA separate, and a single strand molecule of RNA forms a copy of the non-template strand in the DNA, but substitutes U for T in the RNA copy. Now, whether that makes sense or not, it's a bit like when you order a pizza in a restaurant. At this stage of transcription is where the waiter writes down what you want on his pad. And then just as the order on that paper or paper pad or iPad is then translated into the pizza that you ordered in the kitchen, the transcription of the DNA is followed by DNA translation, which is like the chef in the kitchen actually cooking the pizza that you ordered. And these three-pronged molecules here of transfer RNA carry the amino acids, which are the building blocks of the protein, or say the toppings of the pizza, in the right sequence to make a protein, just as hopefully the chef makes the pizza that you ordered. But occasionally, though, just as even the best of chefs can make a mistake in an order, so translation by DNA can make mistakes. Now, up until 2013, we really didn't have any reliable mechanisms to correct such mistakes in DNA function. But now we have a number of kind of genetic scissors, and the most famous one, because it is both accurate and cheap and relatively easy to do, is called CRISPR-Cas9. Now, all of the previous is, of course, a very simplified version to enable us just to grasp the point that for now we are beginning to be able to directly alter the genetic composition ourselves of our own genetic makeup to a limited degree, but this will obviously increase. Now, as with any major advance like this, ethical issues can arise, and here is a list of some of them, which I hope to tackle in following lectures, but the one I'm going to tackle in this one is the issue of genetic determinism. Now, this is a slide of Piccadilly Circus in my youth, when signs were made of neon, and I want to illustrate how genetic determinism is a form of reductionism by borrowing from the illustration that Professor Donald Mackay uses in his book, The Clockwork Image. And Mackay says that genetic reductionism, or any form of reductionism, science reductionism, is called nothing buttery. And so scientists might say that what you see here is nothing but the passage of electricity through a noble gas in a tube. Now, of course, that description is accurate. It won't be accurate today because obviously we use LEDs and all sorts of other things, but you can see the analogy would still apply. Because to say that what you see here is no more than that is completely incorrect. There is a message here. Whether you agree with that message or not is another matter, but it's not just gas in a tube that's conveying the message here. So the scientific explanation is necessary to explain how the message is formed, but it is not necessary to, it's not sufficient to explain it in all its entirety. It is confusing, isn't it? And I'll just get back over that. So it's a necessary but not sufficient explanation. Now, the same nothing buttery is at play in genetics. And here's a classic example from one of the discoverers of DNA, Francis Crick, who discovered it along with James Watson and Rosalind Franklin. And he says in the opening paragraph of his introduction of the astonishing hypothesis, you, your joys, your sorrows, your memories and your ambitions, your sense of identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. Now, bearing in mind what I was somewhat inadequately trying to explain from Piccadilly Circus in the way of Professor Mackay's nothing buttery, you may want to switch the video off at this point and try or pause it to try and determine where in this paragraph Crick is engaging in nothing buttery. What are the key phrases or key words that show that this is an example of the sleight of hand of genetic reductionism? And then when you turn the tape back on again, I will have given you the answer. So pause now if you will. And too late, they are now highlighted in this version. It's the words no more than. So if you got that, very well done. They are a necessary explanation, but they are not a sufficient explanation. Genes are not a sufficient explanation of our awareness, our sense of identity, and our free will, for example. Now, here's another example of this kind of sleight of hand from a recent paper about biology and ideology. Science is not neutral. Scientific method attempts to produce findings which are replicatable and neutral in that sense, but the way in which these are viewed can be far from neutral. So Buchan says here, an increasing number of studies suggest that biology can exert a significant influence on political beliefs and behaviors, suggesting that genes could exert a pull on attitudes concerning topics such as abortion, immigration, the death penalty, and pacifism. So no matter how strongly you may feel about any of these things and think that it is you that is feeling it, actually it's the pull of your genes. And the implication here is perhaps, by using the word pull, the first image that comes to my mind is one of a tug of war. And obviously, if this team here pulls strongly enough against me on the other side, then it is highly likely that I'm going to be pulled right over the line and topple over. So that's not a neutral word, but pull is not a neutral word either. But if you think about perhaps a less immediate analogy, if I, or indeed that whole tug of war team, pull on this anchor chain, it will not actually make any detectable difference in the motion of the boat at all. Now, I know that the physics students among you may have a caveat about that, but I think the general illustration still holds. The boat will not be seen to be moved. And so these constant promotions of the idea that we are nothing but our genetic makeup is not only restricted to genetics. This is a fairly recent book about the neural correlates of consciousness, where Metzinger makes the same point. implicit in all this new data about our evolutionary roots is a new understanding of what it means to be human. And then he goes on to say, strictly incompatible with the Christian image of man. Well, the Christian image of man, for a start, takes account of our free will, whereas absolute genetic determinism certainly doesn't seem to be compatible with that. But it's a point of debate as to whether genetic determinism or a Christian view of the world is a more accurate and sufficient explanation of what we are considering. And of course, if Metzinger were right and it were totally incompatible with a Christian view, it would be impossible for scientists who are convinced Christians like Professor Dennis Alexander to write a book like Genes, Determinism and God, where he demonstrates, I think quite convincingly, that the genetic message itself needs an explanation. He and many other geneticists and scientists who are Christians raised several important problems for any determinist view, for example, that states a causal relation between a gene and trait. If the DNA sequence contains a message, then this presupposes a meaning in that message, and that meaning cannot possibly be generated by the mechanism that translates it. That simply cannot work logically. And furthermore, the DNA has not generated the translation mechanism either, because in order for it to be expressed, it needs that very mechanism. So the genetic message itself requires an explanation, both a final and a causal one. And all these new developments in genomics have, in my view, given rise to even more reasons to be extremely skeptical about genetic determinism. There is evidence now, for example, that genetic effects can be exerted by environmental factors, including smoking and being subject to child abuse. So with the central dogma of the molecular biology that I was taught at school of one gene coding for one protein, that is now increasingly untenable. And that's without even taking into account at all the exploding field of epigenetics, which will take another lecture in itself. So rather than our expanding knowledge of genomic editing pointing towards determinism, I would suggest that it is pointing away from it. The chance of making a designer baby who will grow up to be a virtuoso violinist or the next Einstein are currently simply fantasy on a scientific biological level, let alone a philosophical or theological one. Gene editing, make no mistake about it, is an important tool which is here to stay and can be utilized for either beneficial or harmful ends. But it can never determine how we will use it.